It is my great pleasure to introduce one of the real superstars of the, I don't even know if it's appropriate to call it an emerging field anymore. I mean, it's like an established field of evolutionary medicine. Chris Kuzawa. He's one of my personal heroes. His interests span a wide range of human development. I'm especially interested to hear what he has to tell us about the neurobiological energetic demands during human childhood. He got his PhD at Emory, which has been a real hotbed for human biology for a while now. And you've been at Northwestern for, gosh, a good long time. 12 years. That's my alma mater, so I'm especially proud that Chris has been there. Chris.
Okay, thanks, Mark. All right, and thank you to the organizers for inviting me. This has been great. And I'm going to be talking about human childhood and brain evolution today, building on some of the themes that I think Allison so nicely laid out last night in her plenary. And I'm going to start with a gratuitous picture of my son. Why not? Right? He's about 9 months old here, and he illustrates some things that I think are very unique about humans and how we raise our kids. He's at an age where there's no way that he could go out in the world and actually find food on his own. He'd be more likely to eat nails than actual food, right, at this age. And he's not eating milk. He's eating this specially prepared weaning food that's been cooked and mashed up, and he's being fed by somebody other than his mother. In this case, it's his grandmother. All right, and we take it for granted that this is how we raise our kids, but this is actually pretty unusual. If you compare us to other great apes, we wean a lot earlier. These complementary foods and this early introduction of these foods allows us to wean quite a bit earlier than other species. And there's a pretty good consensus as to why we do this, because if you wean earlier, you start to ovulate sooner, you have a shorter interbirth interval and a higher fertility, right? So the evolutionary advantage of that is pretty clear. But the other piece that you see up here, this very long blue line here, this indicates the age at which first birth occurs, right? So occurs here in gorillas, here in chimps, and here in humans. We really stretch out our pre-adult period. And integral to that is extremely slow growth. Okay, so that's a big part of our early life history. So let's take a look, and we don't really have a good explanation for that. So let's take a little bit closer look at that. Just how slow do humans grow during childhood? I'm going to start with the Kleiber line. Okay, so this is showing metabolic scaling. Think of this as calories per day as a function of body mass. And as you imagine, as you go from smaller species to larger species, total expenditure is increasing, but the exponent here is less than 1. So the caloric expenditure is increasing at a slower pace than the mass. That is, you get an increase in the efficiency metabolically as you go to bigger species. Now, there are all kinds of debates as to why this line takes this form, and caloric expenditure has that relationship with mass. But what it shows for our purposes here is that if you know something's mass, you have a pretty good sense for how much energy it has available to allocate across its various needs. Okay, now given that, and here I'm borrowing from Rob Walker, who's using the cases data from 1978, what this shows is the scaling of growth rate on body mass. And this is growth from birth until about 40% of adult size. And it makes sense that the scaling coefficient, again, is about 0.75, right? Because metabolic processes in general scale with 0.75, so growth is one of those, and it should also follow that regularity. It makes sense that mice and rats and guinea pigs and sheep, they're right on the line. They look like well-behaved mammals, right? And it also, of course, makes sense that the reptiles are down below us because they're cold-blooded. And so for any given mass, a mammal is going to expend a lot more energy than a reptile, and they're going to expend more on growth as a result of that. So that all looks good. What doesn't make a lot of sense is why we are on the reptile line. Okay, we're pretty much right on it. And so if we just take some of these weights right in the range of our body size and plot them as a bar chart, as I've done here, it just gives you a sense for how unusual we are. Right, so these are the other mammals that are in our range. You can barely even see the human line, but when you compare us to other reptiles that are about our size, we look pretty much like a python. So, something weird going on here. Humans grow at the pace of a reptile during childhood. Why is this? Well, we have a partial answer. Herman Ponzer last year and his colleagues in PNAS, they published a nice analysis where they show what we have here is total energy expenditure. Okay? In primates, you can see that it's lower for the same body mass as comparably sized mammals. Right? So this lower energy throughput, they argue, is part of the reason that humans and other primates have slow life histories, that they have slow growth rates and slow reproductive output and so forth. But even when you look within the primates, there's still a lot of variability that's not explained by that trend. And so humans and chimps, for instance, are similarly sized, and yet we grow very slow compared to chimps. So there's still variation that needs to be explained there. And one of the old explanations that's been around for quite a while is that building our brains is so costly that we have to shunt some of the resources that we would normally shunt into growth, we have to shunt it to the brain in order to facilitate that. And so that idea has been in the literature, in the anthropological literature for decades. And there's a kind of intuitive sense in which this makes, it sort of makes sense. Like we really do have big, costly brains. Perhaps we do need to downshift our growth expenditure as a way to compensate for that. But it really hasn't been tested. There haven't been empirical data to try to get at this and evaluate it. And so that's what I'm going to do today. We're going to give some background on traditional explanations for slow and extended human pre-adult growth, tell you what we did to quantify brain energy costs and look at how that plays out across the life cycle, and then consider some of the implications for our understandings of the evolution of the human life cycle, the patterns of growth, and also cooperative care. So we'll talk about all of those at the end. Okay, so first let's just start here. We have brain growth in chimps, pan, versus humans. Of course, we have much bigger brains. And as a group, primates are encephalized, right? They have large brains, but we're quite a bit more encephalized than other primates. So this is a log-log plot. That difference is actually quite large, okay? We're quite brainy. Now, why does this matter? Well, brain is expensive. Brain tissue is extremely expensive. So this is comparing the brain to skeletal muscle per unit gram. I think if you're running a marathon, it's safe to say that this would go up quite a bit, but at rest, the brain is really a dominant energetic organ. And so all of you are sitting here right now probably burning about 100 watts, okay? Heating up the air with that energy. 20 of those watts accounts for your brain, okay? So that's how much your brain is using. And that may seem somewhat dim, doesn't seem all that bright, but if you compare us to other species, most mammals, as you can see over here, devote on the order of 2 to 4% of their resting expenditure to the brain. So the fact that we devote 20 makes us an outlier. And you see the other two primates here for which there were data. Now, the adult, of course, the brain is much smaller relative to the body compared to the newborn or during infancy or childhood, right? So we would expect this ratio to go, this number to go quite a bit up, right, when you look at younger kids. And the old estimates that had been in the literature had our newborns as expending 60% of their total expenditure at rest going to their brains. So given this, if this is even in the ballpark, it seems reasonable that our brains are costly enough that we might, in fact, have to slow our growth a little bit. Now, if that, in fact, was the case, we can come up with some hypotheses about this, right? If our costly brains are requiring a slowing of growth rate, then we should expect an inverse relationship between them. We should expect the ages of peak brain needs to be the ages of slowest growth and vice versa. And so how would we go about this? Well, we could compare, again, this idea of like percentage of our resting metabolic rate. We could look at that, which is sort of saying what fraction of our total energy pie, which is set by our body size, do we devote to our brains? How much does it dominate our body's energy budget? That should give us a sense for how strong the trade-off is between our brains and our weight gain. Now, the problem is that we don't know a lot about how costly the brain is and how this changes developmentally. And the approach that had been taken in the past was to take an estimate of the expenditure required of a gram of brain and then multiplying that through by the size of the brain at different ages and then dividing by the resting metabolic rate of the body, right? So it'll give you this kind of ratio, and you can see how that changes across the life cycle. And so according to this idea, you should have peak percentage of RMR going to the brain at birth when relative brain size is largest, and then gradually it should decline and converge with that 20% figure that I showed you a second ago. And this is, in fact, what Holliday did. in generating the data that I've plotted here. So his estimates are the only that I'm aware of prior to what we've done that estimate percentage of RMR to the brain at various points, allowing us to draw this kind of curve. And so he, at birth, had 87% of RMR going to the brain, and then gradually declining as that ratio of the brain size to body size declined with age. Now, if this is, in fact, the way our developmental energetic profile looks, then there's no evidence that there's been a trade-off between the brain and our growth rate, because here we see that the ages of slowest body growth, okay, so this is a weight velocity, right? So you see the slowest weight velocities are in childhood. This is an age when the brain is in fact decelerating, like it's actually on the decline. There's no indication that there's a peak in brain needs here, as would be required to explain the very slow growth here. All right? Now the problem with this is that the assumptions that went into the making of this brain energetics curve don't necessarily hold, right? Because like I said, basically took an estimate of adult brain tissue, quantified that, and then multiplied it through. So there's an assumption of stability in the cost per unit mass. But PET imaging data tell us that this is not the case. So this is a classic paper from Harry Chugani and his colleagues from 1987. Harry is in fact a co-author on the paper that I'll be talking about, and it's these data that we used. All right? And the way PET works is you provide a very trace level of radio-labeled glucose. And then, of course, as neurons take that up, you have your head in a scanner, and it can sense the rate of glucose utilization in different regions, and you get this kind of heat map showing how much glucose is being taken up. Now, you can't perform PET on normal, healthy kids. You can't just use it for research purposes, because it does involve this trace exposure to radiation. So what Harry did is he runs these PET facilities where kids are referred to him for possible, to evaluate possible tumors and things of that sort, and lesions. And so when kids were referred to him and they got scanned and they looked normal, okay, they didn't actually find anything, he would put them in a data set. And when he did that, and then he went ahead and plotted all of those data, this is what he found. Now this is cortical regions here, and over here, these are the adult controls. So I'm gonna put a dotted line through here. And so what this is showing is the rate of glucose uptake, all right, in these various cortical regions and how that changes with age. Now it's cross-sectional, right? These are not following the same kid through time, it's kids of different ages. But a couple things emerge pretty quickly here. Each gram of cortical tissue uses glucose at a slower rate at birth compared to an adult, right? It's actually below that line, but then it rapidly rises to about twice the level as in the adult by childhood. And here's the actual, give you a sense for this visually. These are some of the PET data. You can see that the brain starts off pretty dim, okay, not a lot of glucose uptake per unit mass, but then it really peaks out somewhere in the 3 to 6 year range, right? And then it declines again by adulthood. And so what's going on here? Well, this has to do with the costs of just synapses, okay? Neuronal synapses. Maintaining neurons and neuronal synapses requires ATP, okay? Sodium-potassium pumps. They're constantly pumping to maintain the gradient, right? And so, and the action potential. And so if you look at the numbers of synapses and how they change with age, you have the period of synaptogenesis, and it varies by region and by function, as we learned about last night in last night's plenary lecture. So early on, you have things like seeing and hearing, and then language, and then higher executive functions, and so forth. So you have this neuronal Darwinist process, right, where you have a proliferation of synapses, and then you have use and disuse dependent pruning, right? So if you don't use particular pathways, those synapses are eventually lost. And so there's a temporary increase in the cost associated with this, right? You've got a peak in the middle childhood years. So these are some classic data from Huttenlocher. There actually are not a huge number of data on synaptic densities in humans, but these are some of the classic ones, showing the increase in synaptogenesis that's occurring after birth, and then you have the synaptic pruning after that, right? Later in childhood and adolescence. So these data reflect the fact, these dynamics that we see reflect these underlying dynamics in the density of synapses, their production and their maintenance, okay? And what all this means is that when we go back to this old chart, it really looks like this is gonna need to be revised, right? First, the idea that 87% is going to the brain at birth is an overestimate, because we know now that each gram of brain actually uses less glucose at birth than in the adult, and the adult value is what was used in calculating these curves. On the other hand, childhood is greatly underestimated here, right? Because you've got this doubling of glucose uptake relative to the adult. So we hypothesized that the curve might look something like this once we take all of this into account. And so that's what I'm going to talk about for the remaining time of my talk. Talk about what the actual costs of the human brain are, accounting for all of this. And then once we do this, do we have evidence for a trade-off between brain energetics and body weight growth rate sufficient to potentially help explain why we slow our childhood growth to the extent that we do? A lot of collaborators on this paper. This was actually funded by an NSF hominid grant, which had various components, molecular, comparative. So this is just one piece of that collaboration focused on energetics. In the methods, I've already alluded to Chugani's data from 1987. Basically, we took this data from the cerebrum, cerebellum, and brainstem, which collectively account for the entire brain. We fit cubic spline functions through those to give us a continuous function of glucose uptake in that region, right? And then what we need in addition to that is to know, so that what this tells us is that at this age, each gram of cerebrum uses that many grams of glucose, right? So now what we need to know is how many grams of that tissue are there, so we can multiply it through and actually quantify the total cost of the brain. And for that, we use volumetric data from the NIH MRI study of normal brain development. Nick Lang was another co-author on this study, and he worked on this study, and he brought those data to the collaboration. That started at 4.5 years, so we had to use other data sources from birth to 4.5 years. And then, because these are different data sources, different size populations, and so forth, we normalized everybody to a common brain and body weight series, Decaban and Sadowski, which goes from birth to adulthood. And when we do all that, here's what we find. So here's glucose use by the human brain, and this is the adult level. Notice that the curve ends at 15. That's because that was the last pre-adult PET data that we had. We decided not to try to extrapolate it. This little hump here, I think, is just noise in our data. I would expect that this curve would just keep going down continuously until it reached the adult level if we had better data and better coverage in those years. Right, so things are peaking out at about 5 years in both males and females. Males are bigger, their brains are a little bigger, and they're using proportionally more glucose. That's what you're seeing here. Okay, so there's just a slight difference. But the key thing to take away from this is that at about 5 years of age, in absolute terms, the 5-year-old's brain is using about twice the glucose of the adult brain. And this is pretty remarkable because the 5-year-old's brain isn't even full size yet, and their body size is about 1/3 the size of the adult. So in terms of a burden on the body and a burden on the metabolic budget of the body, it's pretty severe. And so that's what we look at next. Let's look at this in relation to the total expenditure of the body, RMR. When we do that, it looks like this. So the glucose use of the brain is peaking out as a fraction of RMR, okay, as a fraction of total expenditure, is peaking out at about 4 years of age in both males and females, and it accounts for the equivalent of about 2/3 of resting metabolism. It's pretty remarkable. And down below that, we also looked at this in relation to daily energy requirements, which is something like total energy expenditure. So this also accounts for growth costs and physical activity. And it's very similar, but of course it's a smaller fraction because it's a bigger denominator. Okay, so that's what we find. Now we can start to get at this question that we set up in the beginning. Are ages of high relative glucose use also ages of slow weight gain and vice versa? And I'm happy to say that the answer is yes. So here's the body weights from the Decaban and Sadowski series that I talked about, okay, showing body weight from birth to adulthood. These are the same individuals from whom we have the brain weights. So we fit a curve through that, and then as an estimate of weight velocity, we just calculated a first derivative, which gives you a velocity, right? And so that's what it looks like when you do that. Now I've converted these into SD scores so that we can directly compare them, in a unitless fashion, right? So this shows us the weight velocity, how it changes by age, and this shows us the glucose curve that I've already shown you, superimposed upon that. And it's pretty clear that things are starting to line up. So the ages when the brain is most costly are the ages when growth velocity is lowest. Now we can go one step farther here. And instead of plotting both of these relative to age, we can plot them relative to one another and actually see if there's a trade-off between them. Does weight velocity go down as the brain increasingly dominates the body's energy budget? And we plot them that way. Here's what we find. All right, so this down here is percentage of RMR to the brain. This is the kind of glucose, how much of the body's glucose is going to the brain, and this is the weight velocity, the same thing, okay, both as SD scores. If there was a perfect inverse linear relationship, it would be that gray line right there. So I think we're pretty close. And so here's what's going on. Let me walk you through this. So ignore the beginning here, this little hook for now. We can talk about that later. But between 1 and 2 years of age, okay, this is an age when you're having synaptogenesis and you're having brain growth. So both of those things are increasing the brain's requirements for energy, and it's outstripping, the increase in body size. And so the percentage of RMR is increasing. At that same time, weight velocity is decreasing. And notice that the increase in one is about 1 standard deviation, and the decrease in the other is about 1 standard deviation. So they're proportionate, right? And they're proportionate all the way down, as you can see. At this point here, what we're looking at, this is the point in the life cycle when the brain is most dominant. It's peaked out in terms of its glucose requirements. And it's also the point at which weight velocity is at its lowest. So this is where the peak corresponds with the trough. Okay, that's the sort of the first graph I showed you. And then on the other side of this, we now go in reverse, right? Because now the brain is its full size, and now you're having synaptic pruning. So the brain is actually getting less costly. And at the same time, body weight growth is continuing, right? And so what you see here is that you're getting a decrease in the brain's fraction, the fraction of the body's metabolism going to the brain, and you're getting a corresponding increase in the body's weight velocity. And they're proportionate again. They look like maybe about a half standard deviation in this case. Right? So it looks like a nice linear inverse relationship. So these are the males that I've been showing you. The females are very similar until about 8 or 9, and then puberty takes them off the rails earlier. And then if we do the same thing in relation to total expenditure, daily energy requirements, again, it looks very similar. Okay. All right, so that's what we found. Now let's take a look and kind of revisit what some of this might mean for some of the big questions that we set up. So we've had this idea, we've had a pretty good handle on why we wean so early. We haven't had a great explanation for why we slow down so much. And there was this old idea out there that the brain's very costly and that would require a slowing of growth, but the old data that had been out in the literature didn't really support that. But when we take into account the dynamics in the cost of the brain per unit tissue, it seems to line things up pretty nicely. All of a sudden, that peak is not at birth anymore. It corresponds very nicely with the age of slowest body growth. And when you plot them relative to one another, you see evidence for a nice inverse linear relationship. So I think our findings suggest that the slow pace of human childhood growth reflects a trade-off with the unusually high metabolic needs of human brain development. And from this, I think we also get another hypothesis, which is that trade-offs between the brain and other functions are also going to be peak at this age. The idea isn't that the entire extra costs of our brains have been borne by slowing our growth rate. It's that there are trade-offs probably with many functions, one of them being growth rate. So this is something that's testable. I think this gives us some insights also into the dynamics of growth in puberty. So puberty is something that's it's augmented in us compared to other species. We have a very pronounced pubertal growth spurt. And you can think of as many have written about, this is a deferring of the costs of growth, right? So they're deferred to out here, which is an age when the brain is not dominating things anymore. At that point, you're kind of getting to the adult-style pattern of about 20% or so of expenditure, or 30%, okay? All right, so with all of that, I'd like to end with a closing question, actually two, and explore them in a little bit of detail. Who pays for the extra costs of human brain development, and how do we buffer brain metabolism? And the key point here is that brains are not only costly, but they also have a constant metabolic rate. So whether you're sleeping, you're awake, or taking an exam, your brain is basically requires the same amount of energy. Those action potentials have to be maintained. And so the bigger your brain, the more vulnerable you are in a sense, okay, in needing to meet those costs to avoid brain damage. Now, let's get to the first question first. So who pays for the brain? Well, there's this old idea that Bob Martin published quite some years ago now, early 80s, maternal metabolism hypothesis. And the idea here is that in mammals, the size of the brain is actually constrained by the mother's capacity to supply nutrients in utero, right? So that sort of sets a ceiling and sets the trajectory. And then Steve Lee has done quite a bit thinking about how that might play out in different species, different timings of brain development, and so on. Well, what our data show pretty clearly is that in humans, the peak in brain requirements is well after the point when full weaning from breast milk has already happened, right? So it's not to say that back here, the mother isn't bearing some substantial costs. There are. But the peak, whether you look at it in absolute terms or in relative terms, is at a point when maternal metabolism is no longer directly supporting the offspring. So that's the first point. We'll come back to that. Another interesting point is if you kind of plot here, here's where the brain needs are peaking out, and I've plotted that in relation to the body mass index. So this is kind of how plump somebody is, right? And this is a reasonable marker of nutrient stores. So our needs for energy and the vulnerability of our brains, in a sense, in an energetic sense, corresponds to it at a point when we have our minimum onboard energy reserves. So there's kind of a paradox here. And let's go ahead and take a look at a different point in the life cycle to get some insights into this. So this is my niece Allison. She's probably, I think she's 6 or 7 here. So right pretty close to that point where body fat is kind of at its lowest in the life cycle. Here's what she looked like a little after birth, okay? And baby fat is something we take for granted. We just assume that babies are supposed to be fat. Most of our babies are. But this is actually an unusual trait. So if you look at us compared to other species, this is the first paper I ever wrote as a graduate student trying to think about baby fat and what function it might have. And the literature up to that point had been all about thermoregulation. Okay, so we became hairless, we lost our hair, we needed to compensate by having blubber, right? So this is a thermoregulatory adaptation that allows babies to maintain their heat. Well, if you look in the literature, there's not a lot of evidence for that. And we could go into that. So what I proposed is that we're unusually fat because we have unusually large and energetically costly and vulnerable brains. Seemed like a reasonable hypothesis. And so once you can imagine how I felt about that hypothesis once I plotted these data here, showing that when our brain needs are peaking out, we have our minimum amount of body fat. But I actually think the more as I stared at this longer and longer, it started to make sense to me. Because the nature of nutritional stress actually changes quite a bit developmentally. So what's going on back here, all right, at birth and in the run-up to weaning when we're investing so much in body fat, is infection. That's the main source of nutritional stress in young kids. Infections are common because you're born with a naive immune system. You don't have antibodies at birth. You have to go out and physically get sick in order to acquire the antibody memory that then protects you in the future, right? So kids, they're just very prone to illness. And that's why the World Health Organization differentiates infant mortality from child mortality or under 5 mortality. It's different. It's more about the immune system and about nutrition. Now, why does that matter for our case here? Because infection, the way that it imposes nutritional stress is primarily by reducing appetite, so kids stop eating, they have anorexia, and they often get diarrhea, right, which reduces their ability to absorb nutrients from the diet. Both of these things are cutting kids off from provisioning, right? And it's forcing them to go internal, to rely upon their own onboard battery, if you will, right, to keep alive. Now, by the time they're 4 or 5, their immune systems are up and running, right? And so if there's a nutritional stressor out here, it's actually coming from the environment. It's something about a deficiency in the ecology itself. And so out here, because you're not being cut off from provisioning, the social buffering that characterizes our species comes into play and I think makes that fat unnecessary. And so I think this is another point that comes out of our findings that I want to talk about a little bit. I think they point to the importance of social buffering as a likely prerequisite to human encephalization. And we've already alluded to some of this, allocare and provisioning by kin and non-kin, and cooperation and food sharing. So let's take a quick peruse of some of this, right? So a typical mammal, the mother provides breast milk, and that's exclusively what keeps the child alive until weaning, right? And then they kind of shift to self-provisioning. Most mammals, the father doesn't even know who his kids are. They're totally checked out, right? They're not a part of the picture. But there are species with biparental care, and you see this in birds, and you see it in some mammals, and you certainly see it in humans, right? So fathers are a part of the picture. And even in our studies in the Philippines, 4 years ago, we published a study where we looked at this in relation, looked at testosterone changes, right? So men who had high testosterone to begin with were more likely to become pair-bonded fathers by follow-up 4.5 years later. But then their testosterone, as you can see here on the right, plummeted more than everybody else's, right? And they actually end up lower testosterone. So their findings consistent with like the challenge hypothesis, some of the findings in bird species in which there's biparental care, suggesting that males, human males, have an ability to facultatively shift their priorities away from mating and towards parenting. And then we have built-in biological mechanisms and processes that facilitate that, right? And because we don't see this in other great apes, it seems like it's probably derived and convergent in us, convergent with other species that have this kind of system. So fathers are in the picture in ours, okay? So it's mothers and also fathers, but of course there's a whole cast of characters really that are potentially crucial in raising human children. And it's, the nature of the game here is flexibility. It depends on the society. In the Hadza, you see grandmothers and older women in addition to men who are all kind of contributing to the caregiving unit. Karen Kramer's work in the Maya showing older siblings taking care of younger siblings. It just depends on the setting, right? But if you take this cooperative care system and you combine it with cooperation and food sharing, then you get a pretty high octane strategy of buffering. So this is some of Bruce Winterhalter's work from some time back, showing the coefficient of variation on foraging returns in relation to how many people are being shared with. And as you can see, the variability, the coefficient of variation goes down the more sharing there is, right? And this makes sense because if I go out every day and hunt, some days I'm going to have a kill, and other days I'm going to come back empty-handed, right? So the variability is going to be high. But if everybody pools resources, then the variability is going to tend to even out because every day someone's going to tend to bring something back and everybody gets to eat, right? And this is what we see, if you look at like Gervin's work here on Hiwi and Aceh, this is more or less what human foragers do. And here, this is some data from the Hadza, second to last slide here. So this was an experiment where they were provided with honey sticks, and they just kind of watched where the honey sticks flowed within the society. And you can see there were big gifts and small gifts, and they went according to genetic relationships and friends and so forth, right? But the point is that there's just a lot of caloric flow, a lot of resource flow within the society. And so I think it's that combination of cooperative care and this social metabolism, really, that had made it possible for us as a species to evolve this strategy of having an extremely costly and highly vulnerable brain at an age when we have essentially no battery to keep it going. We don't need it because our social metabolism does it for us. So I think our findings, they tell us something about how costly the brain is. They tell us quite a bit about, I think, the evolution of the pattern of growth and the life history of humans. But I also think when you put all the pieces together and kind of think about it, that it has something to say about our sociality, the nature of our cooperation, and its importance for our overall strategy. And with that, I'd like to acknowledge funding sources, my many co-authors, and please check out our paper if you're interested in this because there's a lot of details in there that I didn't talk about. So thanks.